The following video will teach you about the exciting new field of gene therapy, the differences between gene therapy and genetic engineering, and will demonstrate how chimeric antigen receptor T-cell therapy, also known as CAR T-cell therapy, is a prime example of contemporary gene therapy. It is a common misconception to associate gene therapy with genetic engineering, but it's important to understand what separates them. The University of Missouri Medical School describes the two as such. Gene therapy and genetic engineering are two closely related technologies that involve altering the genetic material of organisms. The distinction between the two is based on purpose. Gene therapy seeks to alter genes to correct genetic defects and thus prevent or cure genetic diseases, while genetic engineering aims to modify genes to enhance the capabilities of an organism beyond what is normal. According to the University of Missouri, the real difference between gene therapy and genetic engineering is how people use them. Gene therapy focuses more on medicine and the treating of causes of diseases, while genetic editing concentrates on elective changes and enhancements to the human body. This video will focus specifically on gene therapy and how it can be used to treat the causes of disease at a cellular level.
A great example of modern gene therapy is CAR T-cell therapy. CAR T-cell therapy is defined by the National Institute of Health as a type of treatment in which a patient's T-cells are changed in the laboratory so they will attack cancer cells. T-cells are the keystone of the body's immune response to foreign pathogens. CAR T-cell therapy is becoming a reliable treatment option for cancers such as lymphoma and leukemia. The process of CAR T-cell therapy occurs as follows. First, blood is withdrawn from the individual, and the white blood cells are removed through an apheresis machine. The rest of the blood is then returned to the patient. The T-cells are separated from the rest of the white blood cells, and the gene for the chimeric antigen receptor, otherwise known as CAR, is inserted into them. The new and improved T-cells are grown and multiplied in the lab and are finally given back to the patient via an infusion. The new T-cells are now able to bind to an antigen on the surface of cancer cells and destroy them.
Genome-wide association studies are used to identify small subsections of DNA that have a statistical relationship with the phenotype of a common illness. In the present, healthcare professionals only consider gene therapy in rare and rather unsolvable cases simply because of the novelty of the subject. However, the future of gene therapy involves ending the stigma against this technology in order to further integrate gene therapy into standard medical practice. Due to the recent completion of the human genome back in 2003, the use of genetics in medicine is highly stigmatized. However, despite these stigmas, the research is becoming more and more convincing, causing the public to begin to accreditize the use of gene therapy. Not only are researchers developing gene therapies for certain sequence abnormalities, but are learning about reconfiguring biological pathways in cell function, protein synthesis, and disease infectivity in order to treat diseases based in genetics. However, such advancements do not come without consequence. In a world where healthcare is not accessible to everyone, how do we decide who gets these therapies, how much they will cost, and where we draw the line at health disparity? Irregardless, the headliners of gene therapy research hope to master this new technology and quickly make it available to all. As gene therapy becomes standard, it's important to remember that the therapy itself is healthy and important for the future of public health. Despite the setbacks and promises associated with gene therapy, research from institutes like the NCBI, NIH, and many others suggests that the positives will manifest over time. Well, whether we like it or not, gene therapy is here, and it will be interesting to observe how this extraordinary technology works its way into medicine and society.
Many other promising types of gene therapy are being developed alongside CAR T-cell therapy, including technologies such as TALEN, ZFN, and CRISPR, which can all be used for genetic engineering as well. The most extraordinary quality of modern gene therapy is its ability to create a specific treatment based on one's personal genome. As these technologies develop, subfields like gene therapy are becoming more prominent due to the rise of Darwinian medicine. Instead of forming a treatment plan based off of treating symptoms and side effects, geneticists have shifted the focus of medicine to one based in evolution. Doctors are now better suited to treat the causation of illness rather than associated symptoms, but this requires analysis of the human genome, its variations, and the correlation between sequence and sickness. Instead of treating symptoms like nausea and headaches, doctors now create treatment plans to cure diseases at the DNA level.